Hello, I'm Heba Sailem. I'm a research fellow at the University of Oxford, and I'm happy to be here today to share my work on phenotyping vascular networks. Vascular networks deliver oxygen and nutrients to every single cell in our body. This complex network needs to be continuously maintained and renewed, and this can happen through a process called angiogenesis, which involves the formation of new blood vessels that allow accommodating the tissue needs. For example, during wound healing, pregnancy, or organ growth. However, if this process happens too much or too little, then this can result in many diseases. So it's very important to study the factors that underlie angiogenesis in order to identify how we can better treat diseases that are associated with this process. Luckily, endothelial cells have an amazing ability to form endothelial networks even in tissue culture. So Kubota et al. Has shown that culturing endothelial cells on a matrigel can result in formation of capillary-like networks even within a few hours. So as you see here, it's like cells scattered on matrigel, and these then endothelial cells start forming connections and network-like structures through chemotaxis and migration. Then after that, this through adhesion, the tubes will mature, the weaker ones will be depleted, and this results in stabilizing the network. So this gives us the perfect tool to study which drugs affect the different aspects of endothelial network formation. Although people use this tool a lot to look at the effects of various drugs, it has been really limited because they will look at only one or two aspects of the network formation. So for example, the total length of the branches or the number of branching points. And this doesn't give us a holistic picture of what the drugs are doing. So what I wanted to do is to develop a holistic approach for profiling vascular networks in order to study the drug effects on the various aspects, and hopefully that will allow us to identify better drugs that can go into the clinic. To do that, I used a data set that was produced by Ayman Zain from while he was at Oxford, where he screened the effect of 1,280 drugs from the LOPAC library on tube formation assay. In his original study, he only looked at the drugs that increase angiogenesis or decrease angiogenesis, which means that we are not characterizing or looking at the different other aspects. So what I've done is to develop an image analysis algorithm that allow classifying the different elements in the network. So first, whether what are the tubes or the branching nodes, and also classify the different tubes depending on their connectivity. So if it is connected from two sides, we call it a segment, or whether it is connected from one side or isolated segment. From that, we can calculate different statistics like the total, the average, or the standard deviation. But there are other aspects that we can extract on the connectivity as well. For example, how connected are the different branches by detecting the different components in the network. Another aspect we can measure is the efficiency of a potential flow. To do that, I used concepts from graph theory. So I measured the shortest path between every two nodes, which averaged give us the estimation of the flow efficiency. Other aspects include orientation of the branches, which can constrained by biophysical laws. We can also use the fractal analysis to characterize if there is specific patterns or the complexity of the network. Voronoi tessellation for branching nodes can provide an indication of the homogeneity of or how uniform the branching points are across the well. In total, I extracted more than 100 features for every drug. And here I'm showing you the visualization of these features in reduced space using Disney. So every point here reflects a drug treatment. And based on this, we can now cluster the drugs based on their effect on the whole range of features. So from this analysis, I identified the drugs that result in similar phenotypes. So we can see that here the control-like drugs, which is similar to DMSO in the yellow cluster. While in the red cluster, we see the drugs that inhibited angiogenesis, so like FGFR inhibitor or FGF inhibitor. The blue cluster was an interesting one because you can see that there is a lot of branching, but if we look closely, it is actually immature. So they are thin and they are really short. So looking at the mechanisms of actions associated with this cluster, I found that it's associated with myosin signaling. So things like myosin light chain kinase inhibitors or ROC inhibitors and other mechanisms of action that seems to also interfere with this pathway. On the other hand, the cluster in purple seem to be really increasing angiogenesis, what we call typical angiogenesis, because it increased the branching and improve the network formation. And this would be like the ideal candidate if we're looking for drugs that improve angiogenesis. Interestingly, what I found that this cluster is enriched for many neurotransmission-related mechanisms. For example, glutamate receptor antagonist or serotonin receptor agonists or GABA uptake inhibitors. And these all are known to affect glutamate. Some of them are known to affect angiogenesis, except for glutamate receptors. So I don't have time to get into the details, but what I was able to do is to use multi-omic approach to validate that glutamate receptors can affect angiogenesis in Alzheimer's patient and correlate with their outcome. So if you are interested, please check the paper, which has been published recently. So in summary, it is important to perform detailed image analysis because it can allow us to identify drugs that result in the desired effect rather than looking at only one feature and ignoring the other aspects. So we can look at branching, mesh formation, network symmetry, and topology. Of course, using multi-omic approach, we can validate what we see in vitro by looking how the targeted genes or targeted processes behave in patient based on patient data. So at the end, I would like to thank the Wellcome Trust for funding my Sir Henry Wellcome Research Fellowship, which gave me a fantastic opportunity to work with different groups. And for this study, it was mainly with collaboration with Ayman El Hajjan. Thank you for listening, and I'm happy to take any questions.